So this way we can represent the multiple linear regression equation, and this equation is used to predict the biological activity of untested molecules. So you can use unlimited time. It is a very good start to predict the bioactivity of untested molecules.
Good evening to the dignitaries, faculty members, and participants. On behalf of the organizing team, I welcome all on the third day of AICTE ATAL sponsored 2-week FDP on computational biology in therapeutics and theranostics. In today's session, there will be lecture by Dr. Firoz Khan sir, who is working as Senior Principal Scientist at CSIR Central Institute of Medicinal and Aromatic Plants, and by Professor Niteesh Purohit sir, Professor in the Department of Electronics and Communication in Allahabad. Dr. Firoz Khan sir will deliver his talk on role of correlation matrix, collinearity, and PCA methods in identification of key chemical descriptors, and Professor Niteesh Purohit
or molecular formula representation which controls the biological activity. So if you see the standard or commercial software, even open source softwares, there are options to have more than 1000 or 2000 chemical descriptors. But whether these 2000
questions will be discussed at the end of the session. Now Dr. Nidhi Mishra ma'am will introduce our first speaker of the session. Thank you.
A very good evening to all and one. So today we have with us Dr. Firoz Khan, who is Senior Principal Scientist at CSIR CMAP Lucknow. He has to his credit several papers, invited talks, more than 100 invited talks. He has received research support from both state and central government funding bodies. He has
or molecular formula representation which controls the biological activity. So if you see the standard or commercial software, even open source softwares, there are options to have more than 1000 or 2000 chemical descriptors. But whether these 2000
project as nodal PI, he has published research articles that are more than 120 as corresponding author, 4 book chapters. He has completed 30 research projects. He has to his credit 1 Indian and 1 US patent. He has organized several training programs. He has organized several conferences, seminars, and workshops as convener. He has been a member of editorial board of Journal of Medicinal and Aromatic Plant Sciences.
Or molecular formula representation which controls the biological activity. So if you see the standard or commercial software, even open source softwares, there are options to have more than 1000 or 2000.
Discovery Society, ATAL University. He is faculty for bioinformatics and computational subjects under GNU and academic CSIR program. He is member of the Society of Biological Chemists, India, Bangalore. He is faculty of bioinformatics, computational biology, and computational chemistry at academic CSIR University. His H index is 35, and he has more than 3,325 citations. His educational qualifications and
or molecular formula representation which controls the biological activity. So if you see the standard or commercial software, even open source softwares, there are options to have more than 1000 or 2000.
And he has done his MPhil from UP Technical University, Lucknow, in biotechnology. Dr. Firoz Khan has been associated with CSIR CMAP since 2007. He has served there as Scientist C, and now he has reached up to the position of Senior Principal Scientist. I welcome you, Dr. Firoz Khan, to this ATAL sponsored FDP, and he has been associated with us for quite a long time now. And I welcome Firoz Khan sir to deliver a talk related to this very topic, which helps you to predict
or molecular formula representation which controls the biological activity. So if you see the standard or commercial software, even open source softwares, there are options to have more than 1000 or 2. Thank you all the organizing committee members. I really appreciate you have given the opportunity to deliver a talk on FDP workshop on computational biology in therapeutics and phenotypics. I hope is of very learning experience with you all the participants and even the online as well as offline. I hope you are learning well. So today's come to the our topic. I have taken on molecule with the lead identification and lead optimization work, which is expert area of my research here in CMAP, CSR CMAP. And I do use these computational methods and techniques, including the machine learning methods, algorithms, or molecular formula representation, which controls the biological activity. So if you see the standard or commercial software, even open source softwares, there are options to have more than 1000 or 2 from the classical lead identification process. It's a very traditional method and very well scientifically validated and answer the question related with how the compounds are so much active. We don't know. We get the activity through the in vitro assay and in vivo assay. We got the activity of the compounds, the small molecules especially. And if you see the drug, already the known existing drug, all are active. But if you see why or molecules are so much active, if you ask this question, you will find that there is no substantial answer. So to come to the point and to answer this question, the only reason is to find the hidden mechanism behind the structure. If you compare or molecular formula representation, which controls the biological activity. So if you see the standard or commercial software, even open source softwares, there are options to have more than 1000 or 2. They are very different with each others, although structurally they are very similar. So it means something is hidden within the compound structure that controls the bioactivity, which is not superficially you can judge by seeing the structures only. So similarly, we can also see the structures which are quite different in structural representation. And if you see the anti-cancer active compounds, and you will find that all molecular structures are very different with each others. But the beauty is that all.
Excuse me, sir.
Yeah.
Sorry for interruption. Actually, slides are not visible.
Yeah, actually, I'm not shared it. Actually, I'm just going through the introduction part.
Oh, okay, sir. Sorry.
Or molecular formula representation, which controls the biological activity. So if you see the standard or commercial software, even open source softwares, there are options to have more than 1000 or 2 is same. So why the similarity is there in the activity? We don't know. To answer this question, we have to use the technique of QSAR to find out the hidden chemical properties which controls the bioactivity. So the today's talk is overall on the QSAR process, and one of the most important steps within the QSAR is chemical features identification. So that is a very tedious task actually. Or molecule to enlighten the participant or give the knowledge to the participant how it is very tough to identify key chemical descriptors controlling the bioactivity. So that's why we have taken a few methods right now or molecular formula representation which controls the biological activity. So if you see the standard or commercial software, even open source softwares, there are options to have more than 1000 or 2 slides. Let us start with the presentation. So are you able to see the presentation or molecular formula representation which controls the biological activity? So if you see the standard or commercial software, even open source softwares, there are options to have more than 1000 or 2000 chemical descriptors. But whether these 2000 or 1000 descriptors are correlated with biological activity and how many actually correlate predictions and develop the model without considering the key component in the process of modeling. So this area comes under the molecular modeling part, and after that we go for simulation and docking studies. So you see we have discussed like three component here: the role of correlation matrix, collinearity, and PCA methods in identification of key chemical descriptors. Or molecular, the definition of the QSR, it is a quantitative way of correlating the structural properties or molecular formula representation which controls the biological activity. So if you see the standard or commercial software, even open source softwares, there are options to have more than 1000 or 2000 chemical descriptors. But whether these 2000 or 1000 descriptors are correlated with biological activity, and how many actually with the see the correlation with the biological chemical properties, you will find there is a correlation between the biological activity and the properties. So which property is more important? So we will find through these three methods basically. Or molecular. Going in detail, I just highlight the why it is necessary to consider the QSR modeling in lead identification. This is an alternative method in research lab, especially the medicinal chemistry lab or drug discovery lab. We mostly involved with the chemist and bioactivity session or molecular formula representation which controls the biological activity. So if you see the standard or commercial software, even open source software is animals. So that's why FDA, you see in this slide, the FDA already mentioned that before submitting any compound for approval as a drug or a lead molecule or a formulation in combination of a mixture of 2 or 3 molecules in pure form, you need to submit the QSR data first. So QSR modeling data basically required very much to show the mechanism behind the activity, and that or molecule which is already covered by European group organization that is called an orchestra group, which suggests a guideline for QSR model development that is also called as REACH guideline. And you see in the middle highlighted part, REACH guideline suggests that four major points should be followed or molecular formula representation which controls the biological activity. So if you see the standard or commercial software, even open source software, property related thing. So whatever the model you are trying to develop, at least follow these four criteria. And the similar four criteria is also mentioned in OECD guideline, which is a worldwide regulatory organization which suggests a standard guideline for QSR model development and other methods also. So if you see the OECD or molecular formula representation which controls the biological activity. So if you see the standard or commercial software, even open source softwares, there are options to have more than 1000 or 2000 chemical descriptors. But whether these 2000 or 1000 descriptors are correlated with biological activity, and how many actually correlated with biological activity, we don't know actually. So to find out the best descriptors, we use some website, REACH website and US FDA website, and see what are the parameters required for prediction of active compounds, toxic compounds, and biotechnology related compounds. Or molecular. Now come to the history of the QSR. QSR is an old method, basically first time used by Corbin Herman Hensch in 1918 to 2011 period, and or molecular formula representation which controls the biological activity. So if you see the standard or commercial software, even open source softwares, there are options to have more than 1000 or 2000 chemical descriptors. So hydrophobicity first time considered as the factor controlling the biological activity. He related this relationship in terms of line equation, which is a multiple linear regression line equation, y is equal to mx plus c, where y is a log 1 by c concentration, and the mx is basically the chemical property independent variable log p, and the other factors are also involved as per as the Hensch equation, substitution constant or molecular coefficients. So these are the first time relationship developed by Hensch in 1969, and you see first time chemical descriptors come into the mind of the researchers. He suggests that hidden property which or molecular formula representation which controls the biological activity. So if you see the standard or commercial software, even open source softwares, there are options to have more than 1000 or 2000 chemical descriptors, mimics the bioavailability parameters. So with this log p value, we can evaluate the parameter of compounds and see if you compare the Lipinski's rule of 5, they have already mentioned, that group already mentioned that log p should be not more than 5. Although some exceptions are there, but standard drug should not cross the value more than 5. So it means very important property it is. Or molecular. So what do you mean by molecular descriptors? These are the chemical properties which are in numerical values forms, and they have a relationship between the biological activity and the different descriptors. These descriptors in regression analysis or molecular formula representation which controls the biological activity. So if you see the standard or commercial software, even open source softwares, there are options to have more than 1000 or 2000, because like identifying the key factors within the chemical properties or the molecular descriptors is a very tough task. If you see the types of the chemical descriptors or chemical properties or molecular formula representation which controls the biological activity. So if you see the standard or commercial software, even open source softwares, there are options to have more than 1000 or 2000 chemical descriptors. But whether these 2000 or 1000 descriptors are correlated with biological activity and how many actually correlated with biological activity, we don't know actually. Chemical properties which comes under electrostatic descriptors. Also the properties which are related with topological descriptors and geometrical descriptors. And lastly, and the recent one is quantum chemical descriptors. So these are molecular orbital related properties, energy related properties, which also found that they have role in controlling the biological activity. So these are the hidden properties within the structures apart from atom representation or molecular formula representation which controls the biological activity. So if you see the standard or commercial software, even open source softwares, there are options to have more than 1000 or 2000 chemical descriptors. But whether these 2000 or 1000 descriptors are correlated with biological activity and how many actually correlated with biological activity, we don't know actually. So to find out the best descriptors, we use some method for descriptor identification, and that descriptor is identification descriptor considered as a chemical feature. So that's why I use the title chemical feature identification using these methods. Now let's see the structure of chemical structure file. If you open any structure file which is based on X-ray or NMR based elucidated structure or submitted in somewhere in the databases of PubChem, Campbell or any other database, you will realize that for different file extension of a small molecule, more or less upper part, structural part is almost similar in nature. I highlighted some of the component here. If you see on the top left hand side, I show the ACD structural file in mole format, which shows the header region as well as after header region. 1 to 3 rows shows the header and 4 to 17 shows the connection table. So 4 to 17 is basically divided again two part. If you see, the major part is atoms coordinate values and the second part is bond related information. So number of bonds are also mentioned in the structural file, and similar structure also found in ChemDraw file also, which is a CDX file format as well as in mole format, any format. So if you open the file, header region already mentioned the type of the file. You can see the definition because first name indicates the molecule name, but second line indicates the program or the software name which developed the structure actually. So you see in the first diagram, you see the ACD lab is mentioned there. It means the structural file is elucidated through ACD program or software, while the lower one figure represents the ChemDraw file, which is representing the atom's coordinate value as well as atom bonds also. And this way it indicates that there is no information about the binding site or the bioactivity related information within the structural file. So how to find these activity relation properties? It is a method which is also called as QSAR method. We use these information in QSAR model and try to find out the relationship between the properties with different properties one by one and find out the potential correlated properties which have a bioactivity relationship. Then we develop the model. So if you see the chemical descriptors file calculated through some commercial software like Discovery Studio, Sibyl or Schrödinger or even the Paddle software which is open source freely available, you will realize the file structure shows in this like manner as is shown in the slide. You see the column represent the different chemical properties while the rows indicate the different compounds in the training set. So these training set compounds are basically the known experimentally known active compounds. So it is a in vitro tested or in vivo based tested data. So you can either calculate it in your lab or as well as you can take it from the databases also or from the literature. You have option. But whatever the molecule you have taken, try to calculate the maximum properties in 5 major categories which I already mentioned earlier in the slide, and try to see whether those properties which have zero value, try to remove that one, and rest property you can calculate it. You can make a correlation matrix. So correlation matrix, when you draw it, it looks like this one. So correlation matrix basically reveal the correlation coefficient value which should be for it, because this correlation matrix among is developed between the descriptors. So it shows the relationship between the one property with the other property. If you see first column represent log p value, while the rest rows indicates the rest of the properties, and those properties which shows coefficient value more than 0.5, you can understand that these are highly correlated. So highly correlated descriptors you can consider as a rejection point, because you are considering the similar type of the property for making the relationship. So out of like similar set properties, you can select any one property. So this is the way we can check the correlation using the matrix for identifying the key chemical factors. For more understanding, I just try to show you that the type of the relationship. There are two types of relationship in regression analysis in QSAR modeling step. One is as shown is that one is dependent variable is represented by dv. IV means independent variable. So there may be possibility that only single property have a relationship between the dependent variable. So that kind of relationship or that kind of equation indicated as a simple linear regression equation or QSAR equation with simple linear regression method. One-to-one relationship indicates. While it is likely very less, but there are so many other examples. If you see lot of properties, at least maximum 5, but there may be possibility that in this slide I show the 4 properties, 4 chemical descriptors as represented by independent variable IV having a correlation with the dependent variable. So 4 line indicating the correlation. So this kind of the correlation may be possible between the descriptors and the bioactivity. Dependent variable is bioactivity. So this kind of the relationship is called as many to one relationship, and this is also called as multiple linear regression analysis or equation you can generate. Now there is also possibility that, as I mentioned earlier, there may be possibility that whatever the properties or chemical descriptors you have identified as key chemical features controlling the bioactivity, they have high correlation between each other. As it is indicated in this diagram, you see IV blocks are intercorrelated with each others as shown by different color in dotted lines, and complete lines indicating the relationship between the dependent variable. So dotted line indications, the dotted line relationship actually showing the almost 6 relationship, while the 4 relationship making with the independent variable with dependent variable. Total 10 relationship considered here. But the problem is that independent variable having a high correlation with each other. This means this relationship is not good enough. So you need to improve it. This kind of like relationship between the independent variable or the chemical descriptors is called as, say it is more than one, so it is called as multicollinearity. So multicollinearity should be avoided. For more understanding, I just shown some of the graph. If you plot this data in a scatter plot, you will find the relationship like this one. First, you see from the left to right, first indicating the linear line in a diagonal fashion. You see this indicate there that these two properties, x-axis or y-axis, have a high correlation with each others. They have positive correlation. But if you see the second diagram and the third diagram, they don't have any correlation between the properties x and y axis. So this way we choose the uncorrelated descriptors and reject the correlated descriptors. More important point I just like to mention here for those who are like willing to do some research work in this area, that always keep in mind that multiple regression is an extension of simple linear regression, and you start with simple linear regression first and then go for multiple linear regression. And the second point is 2 or more independent variables are used to predict or to find out the variance in one dependent variables. And the third point is to remember to avoid overfitting and multicollinearity during the modeling process. Overfitting is caused by adding too many independent variables, while the multicollinearity, as I mentioned, happens due to the correlation between the independent variables. So keep in mind always that these parameters should be checked before developing a model. Once you develop the relationship, it looks like an equation form. It's a line equation, it's a linear equation, and here three variables, independent variables have been used, having a relationship between the y, which is a dependent variable, while it is a line equation since it is a y is equal to mx plus c. And you see, are you able to hear me?
Yes, sir, audible.
Okay.
Yes, sir. Yes, sir.
Yes, sir. Yes, sir.
Message that not audible. Okay. Anyway, I just start with the equation, how to interpret the QSAR equation. So the first value indicates the coefficient value, which is a constant value, which indicates the intercept in the graph representation, while the second x1 and x2 and x3, and they're multiplied by some coefficient value, which indicates the slope of the line. So it shows the correlation where line is indicated by these 4 parameters. One is constant, which is shown in red in color, B0, and the x1 and x2 and x3 are independent variables. So they have indication of slope representation as well as coefficient values. So this way we can represent the multiple linear regression equation, and this equation is used to predict the biological activity of untested molecules. So you can use unlimited time. It is a very good start to predict the bioactivity of untested molecule or virtually designed molecules these days. And you can screen thousands of compounds without going to wet lab and find out the potential compounds without any in vitro testing or sacrifice of the small animals, especially mice, cis albino mice, rat, and like that. So to find out the equation is the beauty of this whole exercise. Now, how to find the key descriptors? I mentioned 3 methods, and one of the most nowadays to converge high and diverse and noisy data to a meaningful data. It reduces the dimension. So that is the beauty of the PCA. Majorly, the aim of the PCA mentioned how the researchers in research labs are using this method.
Excuse me, sir.
Yeah. Yes, please. Thousands. So as it is mentioned, I mentioned for the participant purpose, for their knowledge, they can understand the equation and component also. That it is a statistical procedure. Nowadays, it is considered as a machine learning method. Earlier, it is a PCA is a mathematical method actually, and comes under the linear algebra method. So that's why most of the biologists not use it very well. But nowadays, it is software available nowadays to convert use and find out the results. And principal component basically a standard tool nowadays to converge high and diverse and noisy data to a meaningful data. It reduces the dimension. So that is the beauty of the PCA. Majorly, the aim of the PCA mentioned how the researchers in research labs are using this method.
Excuse
And
me,
you
sir.
can also, yeah. Yes, please. Yeah, Jay Prakash, please. I'm not able to hear you. Jay Prakash Yadav, please.
सर उसमें लास्ट में यक्स थ्री कंपोनेंट जो कि नेगेटिव है से कोरिलेट कैसे करें? अगर हम ग्राफ प्लॉट करें तो वो तो राइट हैंड साइड पे जाएगा सर, तो कैसे कोरिलेट कैसे करेंगे उसको?
बेसिकली ऐसा नहीं होता है। जो नेगेटिव का इंडिकेशन का मतलब है नेगेटिव कोरिलेटेड है ये प्रॉपर्टी x3, और
यस।
x2 x1 पॉजिटिव कोरिलेटेड है बायोलॉजिकल एक्टिविटी से y से।
ओके ओके ओके
इसको
ओके सर।
इंटरप्रिटेशन ऐसे करते हैं कि अगर x की वैल्यू x1 की वैल्यू बढ़ाओगे तो y की वैल्यू बढ़ेगी क्योंकि पॉजिटिव कोरिलेटेड है, और
जी जी
x2 भी पॉजिटिव कोरिलेटेड है। लेकिन x2 और x1 में सबसे ज्यादा इफेक्ट पड़ेगा x2 पे क्योंकि उसकी 0.383 टाइम्स
यस
मैग्नीट्यूड है उसका,
यस
जबकि x1 का 0.014 है। इसलिए
यस
जो चेंज आप कर रहे हो, थोड़ा सा भी चेंज इस प्रॉपर्टी पे करोगे x2 पे, वो बहुत बड़ा इफेक्ट डालेगा बायोलॉजिकल एक्टिविटी पे।
ऑब्वियस ऑब्वियस यस
थ्री जो है निगेटिव कोरिलेशन है, इनवर्स एंड एक्टिविटी। x3 की प्रॉपर्टी अगर आप बढ़ाएंगे तो y घट जाएगा, एक्टिविटी घट जाएगी।
ऑब्वियस ऑब्वियस
And this is the procedure of the PCA analysis. Now come to the aim of the PCA. If you see, majorly we identify the pattern through PCA and to detect the correlation between the variables. This is a best mathematical method, and also the most important is reduce the dimensions. And the important part in QSAR is to find out the chemical properties. So these are the four major aspects of the PCA, and this is the procedure of the PCA analysis. Try to start with standardize the data, align the data, and then perform the singular vector decomposition. And if you know that to identify, it is a process of identifying the eigenvector and eigenvalues. And then after eigenvalue detection, you just sort it into the form of descending order and choose the k eigenvectors, number of vectors, plot the same graph, and then construct the projection matrix. This is the next step. And after that, transform the original data set into a large vector scale so that you can visualize the data. So this way you can easily see the variation. Although I understand that it is very tough to go through the method, those who are not aware of it, but I will try to show the same method in a pictorial representation. I hope you will understand that. Plot the same graph. We know how to plot the two dimension in like MS Excel. It is very easy. We can plot X and Y and then plot the graph. But for example, even we have a software to develop the three-dimensional scatter plot also. But those graphs which are like having a dimension more than 3, 4D, 5D, 6D, even the 110 Ds, 100 Ds, and 1000 dimension, even n number of dimensions. So how you will plot the same graph, similar kind of the graph in an Excel sheet to see the variation? So it is not possible because there are so many dimensions you cannot plot in a Excel software or any softwares. So we have to have a method to converge it into the form of 2-dimension and 3D form, 3D through PCA. So how we can do that? We just try to show you the representation. Like in this diagram, you see there are starting data point x and y, and some data point is already mentioned here as represented by blue color, and perpendicularly just plot the point in the x-axis. You will find the distance between the data points. This distance is basically is called as variance. So in compared to y-axis, x-axis shows very less distance. That means very low variance. So we normally consider higher starting data points. So in this case, we cannot drop the y-axis. Although we showed here that make a cross on the y-axis, but for your understanding, if you converge into the x-axis, we can get the one-dimensional result, but it is not good idea. Now, if we do the same thing with the y-axis, we can plot the data point in a y-axis, and we got the distance in high number with very high distance, with it shows that high variance is there. So the starting data point and the end data points have a high distance. So PCA works well where there is a variation. So always select that axis. So in this case, we can select the y-axis, which is a very good idea to cut down from 2 dimension to 1 dimension. Now there may be possibility that data point may comes in between, and both sides, if you calculate the distance, it will look very similar. Because data point is so close to each other, we cannot middle first more clearly. So we need some kind of magnification, and that magnification can be done through PCA. After magnification, we can calculate the distances and identify the higher distance and then select that axis. So that is the process of PCA. Now how it is done, let's see. In the standardization process, if you go through with the data points, we normally we move the axis in the middle first and then rotate the axis actually. So tilt the axis slightly to align the data point. Now we can remove y-axis, which is x2 due to low variance axis, and only one axis will be there. So this way we can see that if you plot the data point in the y-axis, it look like this one from left to right, you see, and on the x-axis you will see data 100. So you can cut down these 3 initial dimensions or properties into one property. For example, size, number of room and number of bathrooms are highly correlated with each others, and their formation basically is it's a parts of linear algebra. So you can just compare the axis y and x, you see the distance higher in x-axis, this way we select it. So that is the beauty of the PCA. So we cut down the dimension here. Now let us see another example, general example for more understanding about the PCA. There are, for example, housing data is there, and there are 5-dimension data is there. For example, size, number of rooms, number of bathrooms, schools around the crime rate. So you can cut down these 3 initial dimensions or properties into one property. For example, size, number of room and number of bathrooms are highly correlated with each others, and they are formation basically is it's a parts of linear algebra. So it linearly transforms the value from low value to high value. It transforms the low distance value into high distance value. You see in the first diagram, you see in the axis arrow indicating you can club together and make another property which is called as location feature. So this way you cut down 5 dimension to 2 dimension. So in other way, pictorial representation, you can understand that if, for example, there are 2 dimension and you want to convert into 1 dimension, you will realize that housing size and number of rooms can be clubbed together to form a size feature in linear dimension, single dimension. Is in the same thing actually. And the next step is to calculate the distance. How to calculate the distance? Here in PCA, normally we use the matrix method, and matrix formation basically is it's a parts of linear algebra. So it linearly transforms the value from low value to high value. It transforms the low distance value into high distance value. You see in the first diagram, you see in the axis arrow indicated by green color and red color. they have look like very similar in distance. They look very similar distance arrow. But if you stretch down through PCA, you will realize that the value is very different actually. When we stretch the diagram, we find that green color indicates 1 unit while the red color arrow indicates 11 unit at the same time. So these 1 and 11 are basically the eigenvectors and eigenvalues. Go for higher the eigenvalues, mostly selected that one. We select the higher one. So if we go move like on y-axis 1 unit, we have to change 11 unit on x-axis. Go for 11 unit on x-axis. So this way x-axis indicates more significant variation. So that's why you can select the x-axis and reject the y-axis, green color part. Only select the red color part. So eigenvector indicates the direction while the eigenvalue indicates the magnitude. So these values are normally represented by PCA at the end of the calculation, and you have to judge by seeing which value is higher. Select the higher one. That axis will be the final one. So this way we can suggest that those dimensions which look very similar, actually only one dimension is more important. So this way we cut down the features. Same method we use in the chemical feature identification and cut down the number of features. For example, we have like merged 1000 chemical descriptor into one descriptor only. It looks like this one only. So that is the beauty of P1 step calculation. More clearly, overall summary you can see here in this diagram. For example, we have a 5-column table and we want to 2-column table. You can see on the left hand side there is a 5-column table, and after PC analysis 5-dimension plot, 5D plot, we made a 5-dimension plot and cut down to 2-dimension plot based on covariance matrix and eigenstep calculation, and cut down into the two-column table where W1 and W2 indicates the more important properties. So this way we can reduce the dimension, and similarly we can reduce the chemical features. Once we find the leads at the end, that it's a cell-based assay bioactivity data. No target specific information is there. So you have to initially select which type of the activity you are taking from the literature or the database. That is more important part. Try to normalize it, and then normalize means take a logarithm of the activity in a molar concentration form, and then make a relationship, a model validation part. So we use the we use the regression coefficient value, which is represented by Q square or cross validation regression coefficient value, which should be more than 0.5. Always remember, model should be more than 0.6, validation should be more than 0.5. Then it is indication that your model development part is going well, and you will get the good model for identifying the leads at the end. That it's a cell-based assay bioactivity data. No target specific information is there. So you have to initially select which type of the activity you are taking from the literature or the database. That is more important part. Try to normalize it, and then normalize means take a logarithm of the activity in a molar concentration form, and then make a relationship between the regression analysis. Or if we get the activity, we normally go for in vivo testing on small animals as a preclinical trials analysis. So experimental activity in terms of in vitro activity, it may be in different forms. For example, in vitro activity of compound, if calculated in terms of inhibitory concentration on 50% population, I see that it's a cell, then it's indication that it's a cell-based assay bioactivity data. No target specific information is there. So you have to initially select which type of the activity you are taking from the literature or the database. That is more important part. Try to normalize it, and then normalize means take a logarithm of the activity in a molar concentration form and then make a relationship between the regression analysis. For example, if there is a value mentioned as Ki value or Kd value, so Ki means inhibition constant and Kd means dissociation constant. These two parameters basically indicates that it is a data of in vitro assay, but it is a target based. Some target specific protein have been used for testing the small molecules. So this way you have to develop three different model, QSAR model for IC50, Ki value and Kd value. So you have to separate. We normally use the training data set 80% part to develop the relationship between activity and the descriptors. And on the horizontal side, you see on the top, I mentioned the column in two categories. One is on the right side dependent variable, which indicates the biological activity in terms of IC50 or EC50 or KD, KI, any activity form. You have to define any one at a time. 80% part we use in the model development part. This 80% part called as training data, as seen in this slide you see on the top. So top 4 rows indicates the training data set having 80% component. Rest 3 rows indicates the testing data points, which are also experimental one, but they are the part for validation purpose. We normally use the for modeling. We normally use the training data set 80% part to develop the relationship between activity and the descriptors. And on the horizontal side, you see on the top, I mentioned the column in two categories. One is on the right side dependent variable, which indicates the biological activity in terms of IC50 or EC50 or KD, KI, any activity form. You have to define any one at a time. And on the rest 3 column indicates the attributes or the independent variables or the chemical descriptors, we can say. You see as represented by X. You will find that there may be possibility that 4 or 5 descriptors may be possible. So these 3 descriptors, for example, I have identified through PCA method or correlation matrix or multicollinearity analysis, identified through whole axis between the X and Y, and make the relationship between the X and Y. We using the regression analysis, where you will find the line equation which is called as model here. This model is basically a mathematical expression of line equation. So we use this method, this model, for detecting the biological activity of the test data set, which is a lower one, which is represented by X bar. And this X bar basically indicates that these are the properties we will put in the equation and calculate the value. Now that value is calculated or predicted by the model is called as Y double dash, and experimental activity is of this test data set is called as Y dash. You can compare these two values by subtracting with each other's predicted activity and the actual activity. You will find the difference. This difference is called as residual value. Should using any method. If the error value is high, should using any method in the middle, you see regression or classification or machine learning data required for running the or developing a model. So, and while in the unsupervised method, you see there is no requirement of the training data set. So we normally develop the model, unsupervised model, when we do not have any information about the activity. For example, in the case of the COVID, more than 100 data points or experiment. This is a pictorial representation of the whole method, which I explained earlier, that one, you have to submit the training set. Here training set is none other than experimental data. It's in vitro data or in vivo data. So handle with care actually. You need to clean the data and classify the data and then process in the modeling process. Calculate the properties, calculate the normalize the bioactivity, calculate the property, make the relationship using any method in the middle. You see regression or classification or machine learning data required for running the or developing a model. So, and while in the unsupervised method, you see there is no requirement of the training data set. Now come to the machine learning method. Nowadays, it's a very commonly used by most of the data mining researchers. Even I'm using in my lab. So I'm using the supervised method. You see, machine learning is divided into reinforcement method, supervised and unsupervised. Actually, semi-supervised considered in the supervised only, but I just for understanding have taken it. In the supervised method, actually training set required, some experimental data required for running the or developing a model. So, and while in the unsupervised method, you see there is a no requirement of the training data set. So we normally develop the model, unsupervised model, when we do not have any information about the activity. For example, in the case of the COVID, more than 100 data points or experimental biological activity data, more than 100 compounds, then you can go for machine learning method, which is classification method. You can use decision coded the QSAR model for corona inhibitors. And still it is lacking because nowadays we do not have substantial data still to make a relationship. So we can use the clustering method. Those who have published the paper using this clustering or pattern detection method, for example, k-mean clustering or hierarchical clustering or dimension reduction, PC method. And in supervised method, we normally use in our lab, in research lab especially, more than 100 data points most of the time because it works very well for a small data set, and experimental data set is very less actually. Less means less than 100. 100 data points if you have, you can use the regression method. It works well and give you the good results, and error value will be lowest. While if you have more than 100 data points or experimental biological activity data, more than 100 compounds, then you can go for machine learning method, which is classification method. You can use decision tree, Bayesian learning, random forest, KNN method also, KNN SVM. But the problem in that I have used that method in biological data, but the performance is not good enough actually. In wet lab, when we go for calculation, we did not get the promising results. So the error value will be high in these high data set analysis points. So I suggest you, if you have a small lab and if you want to do experiment with minimal budget, you please go through with regression analysis first. Use linear, simple linear and multiple linear, even non-linear method. Any method you can use, and then go for prediction. I'm sure it will work. Out of like 10 compound, you will get activity on 8 compounds or 7 compounds. So the accuracy is very high actually. Data. And while developing any model, especially the QSAR model or QSPR model or the QSTR model, QSAR means quantitative structure activity relationship model, while the toxicity related model also developed, where we use the quantitative structured toxicity relationship model. And some of the crystallographer use the modeling for predicting the chemical properties related with small molecule or peptide. They develop data, and which means quantitative structure property relationship. For example, they want to predict the solubility of the small molecule, and they have a crystallographic data. So they develop the model for QSPR related analysis. Any method use the same procedure as I mentioned here. Always try to follow, collect the data very well, seriously, and clean it. Remove the erroneous data and process it by normalizing it. Develop the model. Select the model. Selection of the model is very important task actually. So if you have a very low data set, select the regression method. If you have a more than a high data set, like more than 100 data set, you select the machine learning method. Start with ANN and then go for SVM and then go for random forest. I'm sure you will get the result at the end. And after that, go for testing and tuning and prediction. That is the rest of the part which normally follow most of the procedure. And you have to mention while publishing it, you have to mention the topic like that only. You use these heading while mentioning the model development part. How you develop the model. This saves the IP, actually intellectual property. You have to show during the model development because you have tuned the model and it works well and doing the prediction well. So it's a very well important to publish it at the earliest so that anybody can use it in a research lab. Do not do the repetitive task again. Now see the application part of the QSAR. So normally in worldwide lab, I just witnessed some of the lab. Even the University of California also, they use QSR for biological activity prediction, cytotoxicity prediction, even the molecular properties prediction also. And this QSR application save the wet lab economy and time. And also most important is it is an alternative method to save the life of small animals. So it is now governed by the government of India also. So government of India is asking to develop alternative method, especially the computational artificial intelligence method to predict the activity or toxicity of compounds. It may be a natural or synthetic or semi-synthetic. So we do the same thing here in CMAP and trying to develop the alternative in silico method to filter out the spurious or toxic compounds from the library. So another advantage of the QSR is to understand the effect of structure on the activity, which is not represented by any other method. Normally we do the in vitro assay, we got the activity and some values, but how it is showing the activity, we don't know the mechanism. So to answer this mechanistic part, we need QSR modeling to answer how it is working actually. We have through this method, we have identified like chemical descriptors, important chemical descriptors out of 1,000 descriptors which controls the biological activity in positive manner or negative manners, as I mentioned in the equation with plus or minus sign. And the most important part is once you develop the robust model and accepted by the reviewers worldwide, you can use this model to predict the biological activity of compounds having a similar kind of series or analog you can synthesize, or even you can virtually design the molecules and check the activity. If it fits in the range, selected range, then it's good. Go for synthesis and spend some money, do some in vitro assay and find out the result. I'm sure you will get the positive results. We do the same thing in CMAB. We work with the chemist and biological scientist people in vitro assay lab, and we get the activity also. And this will cut down the cost of the lab and small animal sacrifice also. You can do the same thing in your lab or wherever you are working or planning to work. Now see the successful example. I'll just list some of the drug as published by some reference in research papers. I just taken the snapshot. For example, norfloxacin, lomrazine, metaconazole, flubufen, they all are like identified through QSR approach. So you can use this approach, and it is very old approach. You see the classical QSR means 2D QSR, two-dimensional QSR approach. Although there is a 3D QSR also, but 3D QSR required 3D structure of the protein with the ligand binding pocket. So when you find that crystallographic data, at least 10 or 15, then it will give the good QSR results, and gives the properties in terms of steric energy. That is also a good approach. I developed the 3D QSR model also, but I did not get the results in vitro assay. So I did not encourage my student to use the 3D QSR. First we go through with 2D, and when we get the 2D QSR model active compounds, then go for 3D QSR. And also at the same time, there is a limitation in PDB database that there are so many missing crystallographic data is there. We did not find actual protein structure with inhibitors compounds bound to with specific binding sites. So that is the limitation part. So it is not easy task to develop the 3D QSR. It requires superimposition of the similar kind of the 3D structures using the protein structural information as well as the chemical structure information. So it's a rational drug designing approach, but it is very tough actually. I suggest you if you want to make a career in this direction, then collaborate with some crystallographer first, then go for 3D QSR. That will be a good idea. In CDRI, CSR, CDRI, they have a good lab, they have a good crystallographer. If you want to do in this career, in this area career, you just try to contact CDRI. At the same time, there are disadvantages of the QSR. I explained most of the positive point, but there are so many negative points also. If you are not aware of the regression analysis or QSAR method or feature identification method, you will get the false positive result predictions and it will fail the whole exercise. Even your project will go down. So please take care. Do not do independent research work. Always collaborate with some chemist and some in vitro assay person. In vitro bioactivity working lab must be there along with QSR model. So that will give you the positive chances to have a good model. So normally it happens false correlation done, even the reflection of the complete chemical properties is missing, even the chemical descriptors may not be reliable enough by the softwares. So these are the limitation of the QSR also. You did not get the right data while developing the model. It's like you're compromising the data. So there are so many successful, that's why I mentioned it here. There are many successful applications, but do not expect 2D QSR work all the time. You need some serious teamwork actually. With this, thank you very much. If you have any question, you can ask. I hope I have completed on time. Dr. Samantha, Dr. Mishra, thank you very much giving the opportunity to deliver a talk.
Dear participants, you can ask your questions to Dr. Firoz Khan. He will be more than happy to answer you.
Sir, I have a question.
Yeah, please.
Sir, do we have sir for this computational datas and all that, should we have the knowledge of programming languages like Python and all that?
No, not required actually. You know the meaning of the machine learning. You use the MS Excel, you use the formula there.
Yes, sir.
Whether you determine that you have used the program there to calculate the summation of the value sum, it automatically
No, sir.
calculates. So
Yes,
that's the beauty. That is the definition of the machine learning. Do some calculation through the machine without any programming. That is called as machine learning. So train your machine to do the task. There are so many program and tools available. You just use it, but you at least understand the principle. So machine learning is a like area where anyone can do the work. All the methods are free, open source. You can use, install your system, and just like MS Excel, you can install it and run the program and it will give the result. But giving the result as a training data set, I mentioned, that is your idea. You have to carefully submit the training data set. Because
sir.
machines do not have an like intelligence. You have an intelligence. You submit the right data, it will give you the right result. If you submit the junk data, it will give you the junk result. Okay.
Okay, sir. Yes, I got your point. But people say that to perform any computational studies, or if you want to go into that field, you should have a knowledge of programmings like Python or Triple C. So I just wanted to be sure.
In the slide, I showed you the results file that are MS Excel. MS Excel is already have all the functions. Use it properly.
Okay, sir. Okay, thank you, sir.
You don't need to use like programming skill. If you want to like develop some method, if you want to develop some tool, then required programming skill. Even if you want to do some large data analysis, for example, you are working in NGS data, next generation sequencing data related with some patients transcriptome analysis or microarray data or the genome data, there you need knowledge of the Linux. Here Linux handle large data set. Linux operating system you need, and especially the Ubuntu flavor. Ubuntu flavor you can use it, and here you need some command knowledge. You can use script also that required Python and R language. Learn the R language. I mostly suggest my student initially to go through with the R language. It is freely available. You can also use it within the R package. There are so many things you can do. Even whatever I showed you, you can do it alone. Even the MS Excel part, you can do it. You can develop the PCA. related model, you can develop the hierarchy clustering, you can develop anything, you can do it. It's a kind of platform. You can just download the library, whatever you want, like you want PCA, download the PC library, install it on the library, on the platform, our platform, and do the task. You only need to submit the input file. It will automatically calculate it and save the output file in a folder. That's the beauty of the R package, even the Python package also. Same way, they both are open source and they are working well. You can, I suggest young participant try to learn and use R package. Initially, it was meant for statistical analysis, but nowadays it is used in all areas. You can generate a very good graph analysis, correlation, network graph also. Anything you can do. Use at least R. R is a good. Python is also good, but R is easy comparatively.
Okay, sir. Thank you so much.
So Dr. Firoz, there are some questions in the chat box. Like, kindly explain role of topological parameter in QSAR.
Okay. Sir, do we, the one question from Sanjana Sakshi, sir, do we fetch the selected compounds directly from other database and perform tasks by this method? Yes, you can do that. One of the most important database is Campbell compound database. You can use it. And other important database is Binding DB, which is developed by the California University. I was also part of that Binding DB project. You can use that one. And another question is that will use of non-linear algo increase the accuracy of the model? No. Non-linear model we normally use for high data set, and it gives not accurate results. For example, if you see the Earth from the space, it looks spherical, you know. So it's a curve fitting model you can use from the space because it covers whole Earth. But if you are sitting on the Earth, like for example, you are in Lucknow or Allahabad, then you will not find curved surface. You look like it's a plane surface actually. It's a linear representation. So whenever the data set is very low, use the linear one and it increases the accuracy. And if you have a large data set, I'm sure large data set always comes in curve type structures or curvilinear type structures. So always use that. At that time, you use the non-linear algorithm. And the next question is, I hope I clear the idea, Srishti. And Sanjana also. If any of you have any question, you can just write it. I'm just reading the chat box. And the next question asked is by Srishti again. Sir, majority of the QSAR paper I have seen were based on linear algo. So how relevant are they on complex biological data? I already explained it that lower the data because experimental data set, in vitro data set, experiment is very expensive actually. So we cannot get all the wet lab data in a one lab. We get data from diverse lab, and these data is basically, if you see in NCVI, PubChem, BioAssay or Campbell, you will find that for a particular type of the activity, for example, Ki value or Kd value, you will find 30 or 40 compounds only, or 90-80 compounds. But that is why we use the regression, because the size is very less. So that is the answer. I hope I clear the point. Yeah, but in future it may be possible that there may be a chance that you have a large data set of in vitro activity database, because present I do not see any research lab data base from India related to biological activity. So we always miss the data. We always take the data from the foreign labs. They have developed the database. We do not have Indian database actually. So it's like a demand of present or future to develop the bioactivity database from all over the, like the funding body is already giving a lot of fund to the different universities and lab, research lab. but I did not get free time to develop the database. But I will try definitely in future to develop a bioactivity database. Another question is that I hope I clear it. Raj K. Prasad, kindly explain role of topological parameter in QSR. Okay, I can take a like analog example. For example, if you want to define a elephant, so you cannot say that only if you touch, if you see the elephant from one side and touch the tail only, you can say that it is a tail of the elephant only. No, if you go in the front side, you will find the other component of the elephant also. So it is better to, if you define some body or some structure, you at least cover all the component from front to back side. So normally we consider all the descriptors, and topological is basically related with topology. So topology should be, as I mentioned earlier, in extended Lipinski's rule of 5, topological parameter should be topological polar surface area should be more than 100, like up to the like 140 angstrom square. So it means some size is fixed to cross the biological membrane to reach from intestine to bloodstream or blood serum. So small molecule should have some specific topology. Otherwise, it will hang in the lipid bilipid layer membrane only. Okay, that's why we have to cover different components during the chemical descriptors process to find out is there any hindrance within the chemical property which restrict the compound in future while testing on clinical trials. So topological parameter is very much required in QSR model descriptor calculation or identification process. I hope I clear the answer because it is related to the topology of the structure. Any other question? Okay. Raj Kumar. Yeah. Biological properties such as IC50 value are assay dependent, may vary lab to lab. Yes, vary. How do you trust the activity while selecting compound from different sources? We normally take the mean value. If you have a lot of like activity, not to favor anyone, select anyone, normalize it. It is better. That's why I'm telling you that always normalize it. It will comes around positive log value around 5 to 13 only. If the log p value is around 5 to 13, then it's good idea that it's a good active compound. There may be chances, I've seen some of the active compounds, they have 4 value also. If you convert the biological activity and P form, P positive value form minus log of biological activity in molar concentration, then it shows the value 4 to 13. 4 to 13 is good enough, but I mostly recommend in my lab 5 is a standard cutoff for good, for 10 micromolar concentration compound. But you can consider 4 also. 4 is a like minimum limit. Less than 4 it is there, it means no activity actually. It may be used in like formulation only or herbal formulation or extract. But as a compound, as a drug, drug means pure compound, single compound. Okay. Formulation means multiple compound, mixture of compound, and extract means thousands of compounds. So we mostly do test the like major yield compounds only. We do not consider the minor ones. So there may be in an extract there are so many thousands of minor compounds which shows the some negative aspect to the extract. Although there is a activity, but when we test the compound in lab, we found that there is a cytotoxicity. It kills the normal cell also. That's why Taxol is not directly used in cancer treatment, or artemisinin molecule did not use directly in anti-malarial treatment. We always use the derivatives, artimol or arti-ether also, or paclitaxel. You heard about the paclitaxel. That is a derivative of the Taxol. Taxol is a phytomolecules. Mostly all the molecules, phytonatural molecules so far discovered as a drug, target the nuclear proteins, nuclear structures, tubulins. The most important part of their target is tubulin. Tubulin is basically maintain the structural representation. So if you damage the tubulin, it will collapse or cell will die. So We normally avoid to give directly toxic compound to normal human being, because at the same time we normally give this Taxol derivative to the cancer patient when there is no chance to survive, because it kills the cancer cell and it saves some time for the patient. Any other question? I hope, Raj, I answered it.
Yeah, thank you very much for the clarification, sir. Yes,
So, Dr. Firoz Khan, thank you very much for such an informative talk. And I hope our participants have got satisfactory answers. So, I thank you, Dr. Firoz Khan, on behalf of organizing committee of CBPP 2022, and we present a virtual memento to you as a token of appreciation. Thank you so much, Dr. Firoz Khan, and we would like to have this connection in future as well. Thank you so much, Dr. Firoz
Sure.
Khan.
Thank you so much, Dr. Krishna. Thank you. It was nice experience. Thank you, participants, all the participants. This is for your workshop.
So,
You can disconnect with the.
yes, sir, you can disconnect.
Okay.
So, participants now will have a brief talk on national importance of national education policy 2020. So we have next Professor Niteesh Purohit as next speaker, and he is a professor in Electronics and Communication Engineering Department at IITA. He is serving as Dean Academics. He has been a national coordinator of Flexible Academic Program Framework, and he is responsible towards implementation of National Education Policy 2020. He has contributed to repeated publications like IEEE Transactions, IEEE Journals, IEEE Signal Processing Letter, Elsevier and Spiritual Journals, and IET Communications in the capacity of author and reviewer. He has co-authored the book COVID War, UK Model, Strategies, Tactics, Impact. Dr. Purohit has delivered more than 60 invited talks in various FDP conferences, workshops, and inspired camps. As principal investigator, he is contributing to many government-funded and consultancy projects, including DST's STI Hub on ICT, Aviral, FAPIS, etc. He has transferred 3D printer technology to Indian telecom industry Mankapur. It is one of his distinct achievements. He is an external member of the Academic Ethics Committee of IIT Kanpur. He is member of Executive Council and Academic Council's Senate Board of Study of many universities and institutes. He is a senior member of IEEE. So we have with us Professor Niteesh Purohit, who will be delivering a talk on significance of national education policy, stating its importance. And now I request Professor Niteesh Purohit to take over and deliver his talk.
Thank you, Dr. Niteesh. I hope I am audible.
Yes, sir, you are audible.
Yes, thank you. So, let me share the slides first. Okay, fine. So, I hope my slides are visible.
Yes, sir, slides are visible.
Okay. So, thank you so much, Dr. Nidhi, for giving this opportunity. And as all know, and okay, thank you. So, there was some echo. Now it is fine. Okay. So, this is an AICT sponsored event, and I just gone through the different topics picked up under this FDP, and I found that these are really interesting, and I was curious that how this NEP 2020 is fits in this particular program. And then Dr. Nidhi told me that this is as it is teaching. And this is something I must say that it is really appreciable, because in whichever domain we are studying, it may be technical or it may be sciences or it may be arts or any other stream, any other discipline. The new education policy announced by the government of India, the national education policy, what we are saying, this is something which is going to have its role in updating almost all the courses, almost all the disciplines. In fact, we have been seeing it as an opportunity. It's an opportunity to rethink the way we are teaching, the way we are designing, the way we are making the, you can say, different resources available to our students. So NEP 2020 is a policy which has put forward a vision. It is just a statement. What should be there in a modern day education system, that you can say. And then the target has been given. It's a 2040. We are saying that this will be fully functional. So a lot of time is given. A lot of thinking is required to be involved, because we are trying to see that there should not be minor changes. And as you can see, the bottom of the slide, in the national education policy itself, section 9.3, it states that huge changes are expected. Amul chul badlav. And that is something what is very, very important, what is very, very bold statement. Just national education policy is not about redesigning, restructuring a few programs, but the education system as a whole, starting from the schooling to the higher education, technical education. In the national education policy, you find that medical and the law education has not yet been explicitly addressed. But when rest of the things will change is on the first priority, then definitely the law education and the medical education is also required to be updated. So what are the expectations of the national education policy that you can see over here, and you can identify that education policy is now emphasizing on this particular aspect, that multiple exits should be made available to the students. As on date, irrespective of the course, it may be a BSc, BCom, BTech, MTech, or whatever it may be. Person enters into the program, and when he completes the program, then only he is worthy of that particular degree. For any reason, if he is living in between, then he is going to get nothing, and it's a loss of 1 year, 2 year, or 3 years of his life, because he studied and because of some reason he could not complete. So the national education policy says that the restructuring should be done in such a way that multi-exit options should be made available. So current systems have been designed with an aim of single exit, and that's why entire restructuring is required. You cannot simply enable multi-exit option in the existing structure of the courses. And whenever we are saying that multi-exit option is to be given, it automatically states that multi-entry should also be enabled. That is, if someone has enrolled in, suppose, the BTech course, it's a 4-year course. For some reason, if someone is leaving after 1 year, 2 year, or 3 year, somehow you enabled it. And after some time, if you want to change his decision, or some important work is there for which he has taken the break, maybe some incubation, he has to start and invest some 1-2 years there. And so the multi-entry option should also be enabled. And just think of it, when we try to actually realize these 2 points, it's a huge change what is required to be done. So multi-entry and multi-exit is something which cannot be enabled in the existing structure. Similarly, the multidisciplinarity, the larger vision stated in the NEP 2020 states that the person should be able to switch from one discipline to another discipline. That is, if suppose if someone has taken the admission, suppose in the BTech with the IT stream, and after 2 years, 1 year, 3 years, or at any point of time he wanted to switch to, suppose ECE stream or some other stream, then currently it is not possible. Once again, he need to go back and restart from the first year. Now, this is something which is required to be enabled. And this is not only about engineering to engineering, one stream to another stream, but it is also stated that actually there should be provision that if an engineering graduate want to go to some arts, history, geography, something like anything, then that provision should be there. Similarly, it is talking about integration of the online education mechanism in the traditional offline. Offline may not be the proper word. You can say face-to-face or classroom teaching or whatever. So this multiple modes stands for this thing. And then a very important part, a very important aspect which has been emphasized in the national education policy is that quality education should be guaranteed more or less to all the students. So this quality education, when we are saying, and then it is something which can be possible if and only if we somehow allow the multi-institute faculties, different faculties available at different institutes, if somehow they can be involved in teaching the students of the other institute, then only it is possible. Because all the institutes cannot have the best faculty available in all the streams. So when we are saying multidisciplinary is to be enabled, and at the same time we are saying that the highest quality of the education should be imparted to the students, then both of this can be guaranteed if and only if we enable the multi-institute capability in the framework itself. Similarly, lateral entry has to be there. Now, fees is also very, very important aspect. It is not categorically mentioned in the NEP 2020, but this is the derived one. And I'm going to discuss more about it. But holistic approach is something which has been categorically defined, very well explained in the NEP 2020, that the approach must be holistic. The students should not be limited to one particular domain. So it is altogether a different approach. We are saying, okay, that there should be the focused teaching. Yes, there should be focused teaching, but there should also be scope of learning life skills, I should say. That is something what is outcome from the holistic approach. The student must be taught about the life skills, little bit of exposure to what are the current requirements of the nation. How can we address it? Some kind of community services or something like this. Yoga may be there, meditation may be there, a lot of things. So how all this to be integrated in all the courses, because all students are our students. And then multiple language medium is also something which is a prominent requirement of the education system. And another feature is the continuous assessment. That is also mentioned in the NEP 2020 categorically, that the examination load, the examination pressure should be released. And it is said that when we are trying to have a seamless assessment in a continuous fashion, so it is also something which is very challenging. The expertise is needed at the level of teachers, because something continuous assessment is something a greater change that is required to be integrated with the education system, because since several years there is a pattern going on. There is a final examination and then everyone is rushing at the examination time. Now this is something which is required to be taken care of, and particularly this part is very, very important at the school level. Perhaps at the college level, higher education, this continuous assessment may have altogether a different way of implementation, but at the schooling level, this is something which requires immediate integration. So having said all these things about the national education policy and their requirements, let me come up to this part of the slide where we are saying that it is the flexible academic program developed by ATAL for its student as a step towards NEP 2020. And yes, we are trying to see that several changes should be there, such that all these features can be made available to the students. So just to let me give you a brief background. So on 29th of July 2020, this national education policy was announced, and we have started working since then, more or less. Several seminars, conferences, several seminars and webinars held. You know, it was a COVID period, so in the virtual mode we held a conference as well. And you can see over here we almost invited and the experts. We almost invited the experts from IITs, NITs, IIITs, the directors and other eminent persons, and yes, experts from the Ministry of Education, Niti Aayog, AICT, chairman, and other persons. A lot of persons we invited, and we just wanted to know their opinion, their vision about the draft that we have been preparing, that is the flexible academic program. And we have incorporated several suggestions into it. And I am very happy to share with you that from this year 2022-23, that is the JOSA 2022, admissions are going on. Our institute has allowed admission of 50 students under this FFP framework, wherein all this features are available. We are offering multi-exit, we are offering multi-tree, multidisciplinary, everything is there. And you can know more about this particular program and the views of the expert. There is a channel that we have created on the YouTube. You can hear the people what they are saying. And if you have some question, that there is a frequently asked question FAQ section is also there. You can just go, and we are extending the support over the chat. It may be on the WhatsApp. You may call. There is a call center. There are experts who will reply you. You can submit your queries, and so everything is there. Everything that you want to know more, I am going to just very briefly give you some information about this flexible academic program. More you want to know, you need to visit here. If you have explicit question, go to the FAQ, submit your question, and you will get the answer. Or ring the pick up your mobile, call that person during working days 10 to 8. You can get the answer for your queries. This is something of first implementation in the India, which is almost offering everything whatever is suggested in the NEP 2020. So there are several other institutes who are trying to do the things in bits and pieces, but at AAIT we decided that no, we wanted to have altogether a new one. So that's a very important part. So what is the larger goal? What is the larger plan that we are trying to see? Our vision is like this, that the institutes of national importance, INIs, that is the IITs, IIITs, NITs, and all these are falling into this particular category. They should act as a cluster. It's a single cluster. And then the entire AICT governed institutes, that is the institutes running under the state technical universities, they may be another consortium, or there may be multiple consortiums here, one consortium belonging to one particular state. And the basic idea is, within the consortium, actually the movement of students, the exchange of faculties, and all this should be provisioned. There should be some mechanism. Similarly here, there should be some mechanism by which the students' movement should be allowed. And what should be the mechanism? We have defined explicitly in the flexible academic program framework. And similarly here, as we said that when we are designing any technical course, we take into whatever it may be, we have to have some HSS courses into this particular. And when we are saying multidisciplinarity, then we have explicitly designed this basket, the non-technical subjects we are saying. So there are baskets belonging to the social sciences. We have entered into the MOU with one of the best institute of the social sciences, that is the GV Pant Institute at the Prayagraj itself. And similarly, we are trying to connect to the right kind of institutes and universities which can prepare customized baskets of the subjects for our students, our FFP students. And we are trying to see that how this can be offered effectively. So that is the idea, that there should be three different, at least, consortiums. There should be provision for the intra-consortium movement, and there should be a provision for the inter-consortium movements too. So that is the larger idea, that how highest quality education should be guaranteed or ensured. So when we say that all this we are planning to do, then it is something which is not possible without a coordinating agency. And this is also something which has been spelled out in the national education policy itself. It is the multidisciplinary educational research universities. So what this should be, this has been properly explained into this. One way is given over there. Another way we are suggesting here is that what a MERU should be, as far as this as a coordinating agency, a nodal agency for these consortiums. So this is one aspect of the FAP program. Now I'm just going to give you a snapshot of what is available. What are the exit points which can be made available? So you can see over here that the course is divided into 8-year span. And then you can see over here that this 8-year is just notional. Instead of year, we are more concentrating on the credits. So flexibility of registering for the credit is available to the students. So yes, there is some upper limit and lower limit. But a student, if someone is registering to the higher number of credits per semester, then this 8 years may be reduced. And if someone is registering less credit, then this is something which will be increased. So this is just notional. The time span is not measured in terms of years. It is earning of credit as per the choice and feasibility of the students. And that's why flexible fee is something which is required to be introduced, the concept of flexible fee. Because if someone is registering only for the 10 credits in a semester time, we cannot charge full semester fee from that particular student. And vice versa is also correct. If someone is opting higher number of credits, then he has to pay the higher fees. And that's why for the FAP students, we have worked out and defined the fees in terms of the credit opted. It's not a semester fees. Semester fees, the notion of the semester fees has gone. So the curriculum, as I said, has been redesigned, and we have ensured that whenever the person is opting for a legitimate exit, that is after earning 40 credit or 80 credit or 120 credits, then the person should learn something which should be complete in some sense. He should be useful in the society. He should be able to serve the society in one way or other. So in terms of certification, the proposed proposition is like this. 160 credit requirement is there for earning the Bachelor of Technology degree, which is also there as per the standard program. In the standard non-FAP program as well, 160 credit is the requirement for earning the BTech degree. In the flexible academic program, what you can say that after earning 120 credit, also you may go for the you may opt to an exit. And there is a provision for 80 credit and 40 credit exit as well. But as of now, for the 2022 batch, we have not enabled this too. Just experimentation is with this. So only after completing 120 credits, one can opt an exit and he can return. As I said already, at any point of time, the person who has opted exit after earning 120 credit, he can re-enter into the system. And we have defined the mechanism for this thing. And after entering, he can continue. So someone may opt and exit after 160 credit, or he may choose to study more. And by studying more, he earns more credits. And then we are saying, okay, after 192 credit, it is there. After 225 credits, MTech degree should be there. And even after he may choose to continue study till he earns the PhD program. So if you just see a person who is entering into the system after the 12th class through the JOSA as of now, he has an opportunity to get the degree of PhD. If he opts full credits and completes this research credit requirements as well, then this span may be reduced. Or someone may go in between works, try his luck in company, in startups. He want to come back, learn something else, and there is something where the multidisciplinarity plays a very, very important role. Someone wanted to go out, suppose someone is studying some IT, go out, work for some startup, and then he over the time learns that he requires the knowledge of mechanical engineering, suppose, then he should be able to come back and learn the mechanical engineering. So that's something which we are trying to provision. As of now, mechanical is not offered. We are offering only two streams. One is in information technology, another is the EC, the electronics and communication. So one more aspect that we are integrated is this thing. Okay. We are saying that under FAP framework, the entire course, the entire program will not have a language medium. So as of now, what is happening that we are saying we are in fact, all the central institutes and central universities, the medium of the language is English. So the logic that we are saying that the entire course is not required to have a medium. We can offer the different courses as per the availability of the teacher in other regional languages or the national language, Hindi language or something like this. So it's teacher's choice. Their teachers may teach individual subjects in different language, whatever they are comfortable, and the students have a choice. You want to study the same subject in a particular regional language medium or the English medium or whatever, depending upon if such an opportunity is available. So in the this is something actually, because when you offer a course, and as I said, the flexible fees and per credit fees is there. So we are saying that there should be altogether a different financial model, and I'm going to discuss a little bit about it, financial model as well. But this is also very, very important, that the medium of the instruction for the different subjects may be different depending upon the choice of the students. So this is something what I was talking about, that we have some targeted skills. Okay. So under the EC or IT stream, when a person earns 40 credits and opt for this, then what should be the taught? What should be taught, and what are the targeted skills? When that is something we need to establish first, and then we need to design the syllabus. So we have done this thing that for the EC students, what are the things that should be known to a person? when he is earning 40 credits or 80 credits and this thing. So there are options. Then there are some very you can say there are different the courses are being offered in a particular way, and this is something what is required to be offered. So now I am coming to the financial part of this Meru, which we are proposing. We are proposing means we cannot create a Meru, it's government job. So what the government should do? That proposal we have submitted. So we said okay, there should be a Meru which government should consider to establish, and then how the Meru should work. That is also there. So in this financial model, what you can see that course fee will be collected from the enrolled students, and then it is suggested that this is the course fee from which the entire system should run. And yes, we can have some donations, we can have some grants as well. And what we are saying, how this should be spent. So we know that teachers are offering the courses, and when we are offering the courses, there is a requirement of teaching assistants, and for running the show, there is a requirement of the backbone support system. So whatever is the income that is being, you can say that is there. So accordingly, expenses can be planned. So we are saying that okay, if per credit fees as of now for 2022 batch, as I said, we are keeping 3,000 rupees per credit. So yes, obviously, so whenever we are entering into the collaborations with other institutes and other teachers, this is the highest figure which is available with us. So accordingly, we are requesting that okay, so you can think of it, and depending upon your requirement, you may keep it little lower than this. And we are getting some faculty members who are saying that they are willing to teach the course for free. So if something like this happen, then actually what will happen? That there are students the registered under this framework who will eventually pay the little less fees as compared to the other students. Or on the other side, you can say that you have some collaboration with some foreign faculties, and they are having a different rate. So that's option will be available. Same subject is available with the multiple fees, multiple faculties, multiple medium of instruction. So it is the choice of the student. So that is the ideal case that we are talking about. Definitely with the 50 intake, we cannot do all this right now, but that is something what has been provisioned, and for which we are working, that something like this should be there. So here I tried to place side by side what is the fundamental principles of the national education policy is written over there, and in the FAP how we have implemented this, or what are the main features of the FAP that you can see from here. So we are saying PhD is the target degree. Now the target has been changed through the JOSA. Students are getting admission in the BTech program, yes, but early exit option is also there, and the PhD option is also. But actually, everyone is getting admission in the PhD. Everyone is expected to study up to PhD. That is very, very important aspect of learning now. then we are saying that there should be a backbone supporting software. I'm just going to talk about it as well, through which this multi-institute movement should be encouraged. And ideally, we are thinking that if after each year students may move from here and there, we have put up a model for it. And I hope that when more institutes will adopt FAP, then we will be able to implement this part as well. But as of now, we are the only institute which is offering FAP, but we are expecting that a few more institutes will join soon. This I have already explained that the number of year is irrelevant now. It is the completion of the credit which is the primary requirement. And the financial model is completely different. The most important part which is there is integration of sports and extracurricular activities, which is also one of the primary focus of the NEP 2020. So we have introduced and we have developed a model as well how this integration should be done. So the holistic education is something which has been ensured after introducing these credits in each and every semester, and this is something which is put in as a one of the requirement. Now, as I said, that all this is not possible if you do not have a ICT platform, and we are developing this. It is in process, but yes, we are trying to see that now everything is digitalized. We know that India is leading the world in the digital innovations, digital solutions. We have seen in the COVID, especially the COVID certificate generation and all these things. And you might have seen newspaper where different institutes, universities are offering the digital degrees online mode. Government of India has also started this academic bank of credits centralized system. So all these are required to be integrated, and we are working towards it, and we are hopeful that it will be done very soon. So this is a very brief introduction. Looking at the time, I am closing here. And just here is with this particular remark that how we design this particular logo for the flexible academic program. So here the design concept is being given. It is the Department of Applied Arts, Faculty of Visual Arts, BHU, which is one of the well-known best institute available. So we have a team and we developed this concept, and then you can see that this is something which is reflecting the vision of the flexible academic program. So let us see, we are starting, and then we can see that where it will go. So I am closing here. If you have any question, then definitely you can ask.
Thank you so much, Professor Niteesh Purohit. Now, participants, please ask any questions, any doubts that you have regarding national education policy, and in future when we will be having flexible academic programs, how to go for them, add to those programs, or how to tackle those programs when we will be hired as faculty in technical institutes. The podium is open to you all. Please clarify all your doubts regarding national education policy, which is about to be implemented in all the institutes. So there is no question from participant side.
Good
Jay
evening, sir.
Prakash Yadav. Yes, yes.
I hope I'm audible.
Yes, sir.
Can you
Yes,
hear
you
me,
are.
sir?
Yes, you are audible. Please.
Yeah. Good evening, sir. I'm Dr. Rajkumar from JP University in Waknaghat, Himachal Pradesh. So I have one question. First of all, very nice presentation. Thank you very much. And majorly it was focusing on the credits and the courses. So my question is about the research, that how do you see NEP affecting the research programs, especially of the PhDs. Currently, we know the situation. There is a huge, uh, we can say, lack of funding for most of the PhD students. So we have to cut short the amount of experiments which actually should be there in the completion of the PhDs. And secondly, my question is that how NEP will uh, 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 affect the faculties, or how the faculty should be ready for implementation of the NEP.
Yeah, yeah, Rajkumarji, your questions are very relevant, and let me clarify this thing. So, as far as you can the research is concerned, what you can notice from the FAP presentation as well, we are saying that a concept of research credits have been introduced, and we are saying as of now it is not explicitly mentioned, but from each and every MTech students, we are expecting that some kind of research exposure uh, uh, should be given to the MTech students as well, not only PhD. So we have explicitly defined that just by earning the credits through subjects, you cannot earn the MTech degree. You have to earn 2 research credits. And how research credits should be earned. For this, we have defined a mechanism. Each and every department of IIIT has defined a mechanism that for attending the conference, which type of conference, for publication, for patents, how much credit should be given. So research is something uh, which has been one of the prime focus of the NEP 2020. If you read the document explicitly and categorically, it has been stated that all these teachings should lead to an environment where the research should be promoted. So there should be no doubt whenever we are creating any program, and when we are claiming that this is something which is in the light of NEP 2020, so all higher study program must focus towards the research. Then only it is the real implementation of NEP 2020. But yes, what you are saying is the availability of the funds. And for this also, let me tell you that we are passing through a transition phase. NEP 2020 envisioned the financial provisions or methodologies in altogether a different way. And since the transition phase is going on, that's why some here and there funding crunch appears to be. But if you just see the policy states that the proportion of the budget which should be spent over the education, NEP 2020 says that it should be drastically increased as compared to. And when the budget is increased, definitely research is going to get a major chunk out of it. So I know here and there what is going on, but definitely I'm not the right person to announce the things. But I can definitely assure you that research is something which is uh, at the center of the entire thought process and entire NEP 2020. And very soon, actually, all researchers will feel it. But at the first, we all need to prepare ourselves, as you rightly pointed out, because education is something which a government cannot change by an order. It is something where not only education, wherever the society is involved, society cannot be changed through an order. So involvement of the faculty, involvement of the teacher, their understanding and their agreement for the change is needed. Then only the change can be done. So yes, we are required to change, as I said, and changing from the examination system to the continuous assessment system is a major change, which requires a lot of thought-provoking and training, I should say, as well, to the teachers. So it is just one aspect which I am saying. But yes, when the entire thing is changing, when the academic framework is changing, teachers are required to be changed, and it must be in a participative way. Okay, so that's what I can say as of now. I hope that I have answered what you was asking for.
Yeah, I think still a lot more debate has to go for a while.
Right.
And of course, there are so many questions. So hopefully, with time, with passing time, we will get most of the answers.
Yes.
So thank you for the nice presentation and answers, sir. Have a
Yeah,
good evening.
thank you. Thank you.
So Jay Prakash Yadav, he has raised his hand. Please ask your question. So in the meanwhile, I would like to ask Professor Niteesh Purohit that will we be having researchers or doing degree programs where the time of a student would be same, but he will be getting 2 degrees.
Actually, there is an echo at your end, so the thing.
Okay, so I will just type the question.
Yeah, you are not audible properly to me. I'm so sorry. So may I disconnect ma'am now?
I think ma'am is writing some question. Yeah,
Okay.
She has written one question. Will we have 2 degree for student in short span?
2 degree means what? Okay, so you are trying. Okay, yes. Obviously, right. It's a multi-degree program. In fact, you can say, because you have earned something and then you have taken an exit and then you can re-enter and complete another degree. So it's just like yes, right. So whenever you are opting for the exit, actually all this remains on the academic bank of credit records. Everything is there. And whenever you are opting for an exit, at that particular point of time, whatever you have earned, the respective degree or certificate will be issued to you. And whenever the student come back and earn the next degree, then definitely that degree will also be issued to you. So it's entirely based on the completion of the credits in a required proportion too. That is also important, I must say. So multiple degrees, yes, these are possible.
Okay, so are there any questions? Because otherwise we will be ending the session. So I think there are no more questions. I thank you, Professor Niteesh Purohit, on behalf of the organizing committee. And now we have a question from Chetan Kumar that students need to spend more time for PhD. So will the PhD be time bound in future according to ATAL?
Okay, so there is a question in the chat box. So should I answer it? Do we have time? Yes, so quickly let me answer it again. Yes, so it is asked that it is a common mindset between professors that the students need to spend more time for PhD, and it drags. How do we answer that? Okay, actually, it's not like this. That the time does not matter. In fact, in any program, actually, it is the output. There are some students. As we said in the FFP, if you just see, we have mapped the research activities in terms of credits, research credits, and you need to complete the credits. So the notion of the time is something which is we want to get away. In the NEP 2020, it is also there. So there are activities which have been listed, and the students, with the consent of the supervisor, he can pick up that what are the activities out of this which should be chosen for earning the credit. There may be some faculty members who may be more inclined towards the suppose conference publications, and he may say, okay, earn the credits through these conferences. Some may say, okay, earn the credit only through the journals. So that is the understanding between guide and the student. But the time as such is it appears to the students that the professors are willing that the students should spend more time, but it is not the time. The time is required for adding quality to the work. That is the primary focus of the professors. So if you can do the good publication in the smaller time, then definitely that degree will be awarded. So time, especially in the NEP 2020 and FFP, I must say, and I can guarantee you that this time has no meaning as such. The credit, earning the research credits is more important.
Okay, so thank you, Professor Niteesh Purohit. Now I think we have no more questions in the chat box.
Yeah.
I would like to thank you on behalf of organizing committee for such a wonderful and informative talk, because our students and young faculty needs to know about NEP 2020, and this has been AICT's initiative that through AICT sponsored programs we make people aware of this policy of the government. So thank you so much, Professor Niteesh Purohit, and I give you, as a token of appreciation, I present before you this virtual memento for this FDP. I will be sharing the certificates as soon as AICTE generates it. So thank you so much, sir, for this wonderful talk, and it was quite informative. Thank you.
Okay, ma'am. Thank you so much. Namaste. Bye-bye.
Okay, thank you, sir.
So, participants, today's program is almost finished. Do mark your attendance in the Google form, and we will be meeting tomorrow, same time, 7 p.m. Tomorrow we will be having two talks. One is on bioengineered human tissues and organs, the way forward in healthcare. That is the domain of upcoming healthcare. And then we will be having a talk on psychology of teaching and learning, which is based on when you take up teaching as a profession, what psychology you have to keep, and you have to learn the psychology of students to make them learn. So both the talks would be interesting. Please do join us on time. Thank you so much.